Cancer mice. What would happen if cancer, a disease we usually associate with humans, could also affect a mouse? In the 1980s, scientists at Harvard University conducted an experiment to answer that question. Researchers inserted a cancer-triggering gene, known as an oncogene, into a mouse's DNA. This gene was programmed to remain active for the mouse's entire life, making it extremely prone to developing tumors. Physically, these mice looked completely normal. However, genetically, they were engineered to develop specific types of cancer, such as breast cancer, lung cancer, and brain cancer. Some of the mice carried genes that actively triggered cancer, while others lacked tumor suppressor genes that normally prevent uncontrolled cell growth. As a result, cancer could develop naturally, without exposure to chemicals or radiation. The outcome was clear. These mice had shorter lifespans than normal mice, and tumors grew rapidly throughout their bodies. However, there was a critical benefit. These cancer mice allowed scientists to study how cancer develops and progresses, and to test cancer drugs, immunotherapy, and gene therapy before they are used in humans. This research has had a major impact on the international medical community. GloFish. Among the many experiments that have created so-called mutant animals, this one is often considered nearly perfect. This experiment was carried out on zebrafish, a small fish species known for its unique characteristics and natural color variations. The creation of GloFish began by inserting a gene that produces fluorescent protein into zebrafish embryos at a very early stage of development. These genes were taken from jellyfish and sea anemones, organisms that naturally produce glowing proteins. Once the genes successfully integrated into the fish's DNA, the fish continued to produce fluorescent protein throughout its entire life, causing its body to glow when exposed to ultraviolet light. In terms of appearance and behavior, GloFish show no significant differences compared to normal zebrafish. Their body shape, swimming behavior, ability to reproduce, and even lifespan all remain normal. Because of this stability, the research was officially recognized around the year 2000, after it was proven that the trait was genetically stable and could be passed down to future generations. However, the experiment also sparked controversy. These fish are now widely sold as ornamental pets. Featherless chicken. If chickens are usually known for their beautiful feather patterns, this one is very different. This is the featherless chicken. These chickens were developed in Israel by researchers at the Hebrew University of Jerusalem. The original goal was relatively simple: to improve poultry farming in hot climates. Scientists believed that chickens without feathers might tolerate high temperatures better, conserve metabolic energy, and reduce the cost of feather removal during processing. Visually, these chickens clearly look unusual, almost mutant-like. Their bodies appear mostly bald, with some individuals growing only small amounts of feathers. Beyond appearance, serious problems began to emerge. Without feathers, the chickens lost their natural protection against the environment. They became more easily stressed and extremely vulnerable to injuries, skin irritation, insect bites, as well as bacterial and fungal infections. As a result, the research was not expanded and was eventually discontinued. Spider goat. When you hear the name, do you imagine a goat turning into Spider-Man? This is the spider goat, a unique scientific experiment involving goats, where researchers inserted a spider gene that produces silk protein into a goat's DNA. At first, the experiment may sound extreme or even crazy, but it was carried out for a practical purpose: to produce synthetic spider silk, a material known for being incredibly strong, elastic, and lightweight. This research was conducted in the United States. Physically, these goats look completely normal, just like any other goat. The real difference lies in the milk they produce. Their milk contains spider silk protein, which can later be extracted and spun into fibers. What makes this even more surprising is that spider silk is known to be stronger than steel by weight and has potential applications such as lightweight bulletproof vests. However, there is a downside. This experiment removes the goat's natural maternal function because the milk produced is not recommended for consumption, as it contains spider silk protein rather than normal nutritional milk. Dolly the sheep. If we were to count her age today, this animal would be nearly 30 years old. This is Dolly the Sheep, a sheep created through cloning in 1996 at the Roslin Institute in Scotland. Her wool, body posture, facial structure, and daily behavior were all completely normal. In the history of science, this experiment is recognized as the first successful cloning of a mammal. Dolly was created using a technique known as somatic cell nuclear transfer. In this process, the nucleus from a mammary gland cell of an adult sheep was removed and inserted into an egg cell from another sheep. The egg cell was then stimulated with an electrical pulse. This cloned embryo was implanted into a surrogate mother, eventually resulting in the birth of a sheep that was genetically identical to the original cell donor. Unfortunately, Dolly lived until 2003. She died from lung disease and arthritis. Zorse. A zorse is a hybrid created by crossing a zebra with a horse. The result is a unique blend of both animals. It usually has zebra-like stripes on part or all of its body, a slimmer horse-like build, and stronger endurance with more wild instincts compared to a regular horse. Because horses and zebras have different chromosome counts, zorses are almost always sterile and often experience reproductive issues, making natural breeding extremely difficult. On average, a zorse can live around 20 to 30 years. Today, zorses can be found mainly in Africa and North America, especially in captivity or conservation centers. Interestingly, there are estimated to be over 1,000 zorses worldwide. Ironically, many experts argue that breeding zorses offers little conservation value, since they're often produced for circus shows rather than for wildlife preservation. Geep. At first glance, this animal looks cute, but confusing. What should you call it? Is it a goat, or is it a sheep? This is the Geep, an animal created through a biological technique that combines two different organisms into a single body. The Geep is formed by merging a goat embryo and a sheep embryo at a very early stage of development. The result is a single individual that is neither a pure goat nor a pure sheep, but a genetic combination of both. The distribution of genes is uneven throughout its body. In some parts, such as the head or legs, a Geep may display goat-like characteristics. In other areas, such as its wool or overall body structure, it may resemble a sheep. The purpose of this experiment was not to create a strange new animal, but to answer fundamental questions in developmental biology and immunology. Specifically, why the immune system does not always attack foreign cells. Liger. The liger is a hybrid animal created from a male lion and a female tiger. This crossbreeding doesn't happen in the wild. It only occurs through human intervention, typically in captivity or in zoos. From its parents, the liger inherits an enormous body size, often growing even larger than both pure lions and tigers. It has the muscular build of a lion, combined with faint tiger-like stripes across its coat. Its behavior is also a mix. Ligers show the social tendencies of lions, but they also keep the solitary instincts of tigers. Because of genetic incompatibilities, not all traits from the parents appear, and male ligers are almost always sterile. Fun fact, there are about 52 zoos around the world that keep ligers, with a global population of more than 120 individuals.